Dear student, I am Dr. Prakash Mungli, professor in biochemistry. So in this video of electron transport chain, I am going to explain you all about inhibitors and uncouplers of electron transport chain, brown adipose tissue and reactive oxygen species and antioxidant molecules. Let's jump into this. Inhibitors of electron transport chain are shown in this particular figure here. So we have complex 1, complex 2, complex 3 and complex 4 inhibitors are shown, and also uncouplers are also shown in this particular figure. Let's look at complex 1 inhibitor. They are pyridine A, amobarbital and rotenone. So all these molecules, like that is pyridine A, amobarbital and rotenone, they inhibit transfer of electrons from NADH coenzyme Q oxidoreductase complex, that is complex 1, into coenzyme Q. So they are referred as complex 1 inhibitors. And then we have two inhibitors for complex 2, that is carboxin and thionyl trifluoroacetic acid, TTFA. Now complex 3 inhibitors are British anti-lewisite and Antimycin A. So what actually they do? They are going to inhibit a transfer of electrons from coenzyme Q to complex 3. So that's why they are referred as complex 3 inhibitors. And complex 4 inhibitors, we have hydrogen sulfide, carbon monoxide and cyanide and also sodium azide. Now uncouplers, what they do? So uncouplers, they are going to uncouple electron transport from phosphorylation process. Thereby, they decrease ATP generation while allowing electron transport chain to continue. There is a molecule called oligomycin. Oligomycin, it is going to bind with ATP synthase, especially F0 subunit in the ATP synthase, and thereby it acts as an uncoupler of electron transport chain. A molecule called malonate. Malonate, it is going to compete with succinate for succinate dehydrogenase enzyme, thereby it decreases the formation of FADH2, which has to enter into complex 2 of electron transport chain. Doxorubicin, it is an inhibitor of a mobile complex in the electron transport chain, and that is coenzyme Q. Now let's move on to see what is the mechanism of cyanide toxicity on the electron transport chain. Now the cyanide, it is an inhibitor of cytochrome AA3, that is complex 4 in the electron transport chain, and also note that complex 4 is called as cytochrome C oxidase. Now note that complex 4, it has 4 inhibitor as such, that is cyanide, carbon monoxide and sodium azide. Out of these, cyanide and sodium azide, they are going to bind with ferric iron of heme A3, whereas carbon monoxide will bind with the ferrous iron. So here cyanide, which is inhibiting cytochrome AA3 by binding to the ferric iron. So what are the sources of cyanide? So cyanide, it will come from fumes from polyurethane, that is house fires. And also cyanide will come from as a byproduct of a medication called sodium nitroprusside. Cyanide is also a part of automobile exhaust, and the cyanide in the air, it will be present as hydrogen cyanide, whereas cyanide in the water and soil, it will be present as a sodium salt of cyanide. Acetonitrile is one of the source of cyanide toxicity. Seeds of kernels such as those of almonds, pits from stone fruits, apricots, peaches, plums, cherries, sorghum, soya beans, spinach, lima beans, sweet potatoes, maize, millet, sugarcane, apple seeds, orange seeds, cassava, that is tapioca, and bamboo shoots. All of them, they contain a little bit quantities of cyanide. Now what is the treatment for cyanide toxicity? Amyl nitrate along with sodium thiosulfite is an antidote for cyanide toxicity. So this amyl nitrate, whenever it is infused, so the amyl nitrate, what it does, it is going to oxidize the ferrous iron present in the heme of hemoglobin into ferric iron, thus converting hemoglobin into methemoglobin. And this 𝐒𝐲𝐵, it is going to, it has got higher affinity for cyanide, and it's going to bind with the cyanide and become 𝐶𝐻 and that will be excreted out of our body. So let me write down the mechanism here. So amyl nitrite, it is going to oxidize a hemoglobin which has got ferrous iron into 𝐒𝐲𝐵. And 𝐒𝐲𝐵 has ferric iron. So hemoglobin is converted into 𝐒𝐲𝐵, and this 𝐒𝐲𝐵, it has got higher affinity for cyanide. So it will bind with that and become 𝐶𝐻 and this 𝐶𝐻 is excreted, and that's how cyanide is taken out of the system. So that is how amyl nitrate, it is acting as a antidote for cyanide toxicity. And also cyanoKIT is used in cyanide toxicity. That is, cyanoKIT is hydroxycobalamin, and this hydroxycobalamin, it can bind with cyanide, become a cyanocobalamin, and that's how cyanide can be taken out of the system. Now let's look at mechanism of carbon monoxide toxicity on the electron transport chain. So the carbon monoxide binds to the ferrous iron in the heme of cytochrome AA3, but it binds less tightly than cyanide. So the sources of carbon monoxide toxicity here, it is improper combustion of organic fuels like propane gas heaters or gas grills, vehicle exhaust, tobacco smoke, house fires, methylene chloride based paint strips. So all these are sources of carbon monoxide. Now the symptoms include headache, nausea, tachycardia, tachypnea, lips and cheeks turn cherry red in carbon monoxide toxicity, respiratory depression, coma results in death if not treated by hyperbaric oxygen. Now let me explain you uncouplers of electron transport chain. So what they do? Uncouplers of electron transport chain, they uncouple electron transport from phosphorylation process. So they allow free flow of protons from intermembrane space through the inner mitochondrial membrane into the matrix. Let me explain you how exactly that happens. So here is the inner mitochondrial membrane where electron transport chain is located. So I am going to write complex 1, complex 2 and complex 4 here, because complex 1, complex 2 and complex 4, these are the complexes that will pump protons from the matrix of mitochondria into intermembrane space. So I am going to write outer mitochondrial membrane here. Now when the electron transport chain is running, so the protons are pumped into the intermembrane space from the matrix side. So as more and more protons are building up here, so the proton gradient is building up. So at that time, these protons, they will come back into the matrix side through ATP synthase. So now because of the proton gradient, so they flow into the matrix side of mitochondria. So while this happens, so ATP synthase is going to phosphorylate ADP with Pi to make ATP, and that's how ATP generation. So there is a phosphorylation process going on when electron transport chain is going on the other side. And also note that in the fourth complex, oxygen molecule, an atom of oxygen, it is used and made as water molecule, H2O. So electron transport chain here, it is coupled with the phosphorylation process, and that happens only because inner mitochondrial membrane is strictly impermeable to protons. So only way that protons can come back into the matrix side is through ATP synthase. What uncouplers will do? Uncouplers, these are ionophores, and what they will do is they are going to insert a pore in the inner mitochondrial membrane. Because they are hydrophobic molecule, they act as a ionophores and they allow free flow of protons through this ionophore. So basically now what happens, these protons, so they come through these ionophores into the matrix side without really going through ATP synthase. So because of this, what happens? So the proton gradient is not built properly so that protons are flowing through the ATP synthase. So that process decreases. So in the less and less protons are flowing through the ATP synthase, so the phosphorylation will also decrease, and that means ATP synthesis will decrease. Although just to build the gradient, so electron transport chain will continuously go on. So they are going to break oxidize NADH into NAD plus FADH2 into FAD plus and continuously pump protons into the intermembrane space trying to build the gradient, but the gradient is not built because the protons are leaking through these ionophores. These are the uncouplers. So what they are doing, they are allowing electron transport chain to continue, but they are not allowing sufficiently phosphorylation of ADP to make ATP by ATP synthase, because proton gradient is not built, because protons are leaking through uncouplers. So that is why these molecules are called as uncouplers, because they are going to simply uncouple electron transport from the phosphorylation process. Electron transport is going on continuously. In fact, more and more NADH is oxidized to NAD plus, FADH2 is oxidized into FAD plus, electron transport chain is running, oxygen is consuming, the respiratory rate is being increased to provide oxygen, but proton gradient is not built because there is a leakage going on in the inner mitochondrial membrane, and this is why phosphorylation in the ATP by the ATP synthase to make ATP is not going on, and that's why there is decrease in ATP here. So phosphorylation is uncoupled from the electron transport. That's why these molecules are called as uncouplers of electron transport chain. So they are allowing free flow of protons across the inner mitochondrial membrane and thereby do not allow gradient to be built. So the energy from flow of protons is simply released as heat because it is not tapped as during the phosphorylation process. So overall, as the protons are leaking through these uncouplers, so energy is just released as heat. So that increases the body temperature during uncouplers. So whenever uncoupler proteins are acting here, so body temperature, so there will be increase in the body temperature, there will be decrease in ATP synthesis, electron transport chain is running faster, that means there is increase in oxygen consumption. Oxygen consumption increases and there will be more and more NADH being oxidized to NAD plus, FADH2 oxidized to FAD plus, and because of this what happens, there is a higher consumption of acetyl-CoA in the TCA cycle, higher consumption of glucose to make acetyl-CoA. Overall, that increases basal metabolic rate. So because of the increase in the basal metabolic rate, so overall body temperature further increases. So during uncouplers, effect of uncouplers is increase in the respiratory rate to provide oxygen, increase in the basal metabolic rate to provide the substrate for TCA cycle, and then NADH over NAD ratio is decreased, FADH over FAD ratio decreases, ATP synthesis decreases. Overall, electron transport is totally uncoupled from the phosphorylation process. Now, what are the examples for uncouplers? So, examples for uncouplers can be grouped under physiological uncouplers and chemical uncouplers. Let's look at physiological uncouplers first. Physiological uncouplers are uncoupling proteins which are embedded in the inner mitochondrial membrane in different tissues. These uncoupling proteins can be of uncoupling protein 1 to uncoupling protein 5. Uncoupling protein 1 is referred as thermogenin, and that is found in the brown adipose tissue, and the brown adipose tissue is usually found during neonates like newborn, and as the growth and development is going on, brown adipose tissue slowly quantity of it is decreases. And the thyroid hormones indirectly they act as uncouplers of electron transport chain. Now coming to the chemical uncouplers. Chemical uncouplers, these are being lipid soluble molecules. They act as ionophores and they carry protons across the inner mitochondrial membrane into the matrix side of mitochondria. So the examples of chemical uncouplers, we have aspirin, salicylates, 2,4-dinitrophenol, dinitrocresol, pentachlorophenol, and succinylcholine, and several other inhaled anesthetics. They can act as uncouplers, chemical uncouplers of electron transport chain. Now, the types of uncoupling proteins that are found in variety of tissues, that is uncoupling protein 1 to uncoupling protein 5 can be found in our tissues, which will in to varying degrees they can uncouple electron transport chain, thereby release some of the heat that is needed for the maintenance of the body tissue. So the UCP1 is a thermogenin present in a brown adipose tissue. We are going to go into little more detail on this in the subsequent slides. Most cells in our body will have uncoupling protein 2, skeletal muscles will have uncoupling protein 3, and nervous tissue will have uncoupling protein 4 and uncoupling protein 5. So the expression of uncoupling proteins in our tissues will make electron transport chain inefficient, because protons will be leaking through the inner mitochondrial membrane. That means a tissue or the cell need to spend more substrate to make same number of ATPs otherwise if the electron transport chain was more efficient. So genetically some people will express more uncoupling proteins in their tissues, and that's why these people they need more food to make the same number of ATPs, and that indirectly helps them to not to gain weight, because they will burn most of the food stuff that they are eating, because the metabolic rate in them, basal metabolic rate in them will be much higher compared to the other person whose electron transport chain is more efficient, because they don't express much of the uncoupling proteins in their tissues. So any person who is expressing more uncoupling proteins in their tissues, so they will have higher basal metabolic rate, and generally they don't put on more weight compared to other person who is not expressing much of the uncoupling proteins in their tissues. So their electron transport chain will be much efficient. That's why even if they eat less food, they will be making more ATPs. So this is the genetic difference that we can see in individuals. Now let's move on to see a little more on brown adipose tissue. And this brown adipose tissue is generally seen in the interscapular areas. It is seen around the organs like kidney, heart, and all that. Usually brown adipose tissue is seen in higher quantities in newborns, and as the development goes from childhood to adolescent, so the quantity of brown adipose tissue will decrease. Now, whenever a person is exposed to cold temperature, or like a newborn, when the newly born fetus, so coming out of the mother into the colder temperature, because mother's womb has 37 degrees centigrade. When the birth happens, neonate is coming out into a relatively colder temperature, which may be 24-25 degrees centigrade. So how to adapt to this colder temperature? One of the way is to produce more energy through brown adipose tissue, and that is called as non-shivering thermogenesis. So what happens in the non-shivering thermogenesis? Whenever a neonate or a person who is having a brown adipose tissue exposes to cold, so what happens? So there will be stimulation of sympathetic nerves, and this sympathetic neurons releases norepinephrine, and especially when it is binding to the beta-3 adrenergic receptor. And because of that, there will be activation of lipoprotein lipase enzyme, and that lipoprotein lipase, it is going to break triacyl glycerol into free fatty acid and a glycerol molecule. And the free fatty acid, it is acting as a positive modulator on thermogenin, and it is going to close the thermogenin. So when the free fatty acids are released, so this free fatty acid, it is going to open the thermogenin, and when the thermogenin is opened up, so that makes electron transport chain inefficient, because the protons are starting to leak through thermogenin from the intermembrane space. They start to come into the matrix of mitochondria. So basically, opening of thermogenin through fatty acid will make electron transport chain inefficient, and the protons that are leaking through this thermogenin towards the matrix side coming from the intermembrane space, so it causes release of heat, because energy is not captured by the proton gradient that is trying to build there. So the heat that is produced here is trying to maintain the body temperature, upon exposure to cold. So this is the response of our body upon exposure to cold, and this type of heat generation in response to beta 3 adrenergic receptors which are activated by cold is called as non-shivering thermogenesis. So since electron transport chain becomes inefficient here, so when that leads to accumulation of ADP or accumulation of GDP, these are adenosine diphosphate, guanosine diphosphate, which indicates lack of energy in the system. So accumulation of ADP and GDP, at that what they will do is they will go and bind to thermogenin and they will close the thermogenin. When the thermogenin is closed, so protons are no longer leaking, so that means electron transport chain becomes more efficient, and now electron transport chain is able to make more ATPs, and thereby it is going to bring up ATP levels in the tissue. So like this, physiological changes will be going on in our body, and whenever there is a need for heat generation, so heat can be generated through electron transport chain. Of course, heat can also be generated by running metabolic reactions, but this is another way of generating heat in our body. So all those points that I have written here, so the location of brown adipose tissue, it is there in the interscapular areas and around the organs, and brown adipose tissue, it is participating in non-shivering thermogenesis. That's why brown adipose tissue is also referred as thermogenic organ. And the brown adipose tissue has uncoupling protein 1, which is also called as thermogenin, and the primary stimulus for thermogenin is stimulation of beta 3 adrenergic receptors which are releasing norepinephrine. So uncoupling protein 1 is activated by fatty acids and it is inhibited by purine nucleotides, that is adenosine diphosphate and guanosine diphosphate. And thyroid hormones, they act indirectly in keeping thermogenin in open conformation, because thyroid hormones, they work through norepinephrine stimulation, and when norepinephrine increases through beta adrenergic receptors, it is going to keep that thermogenin open by increasing fatty acid levels. And also the thyroid hormones, they will increase the expression of sodium potassium ATPase over the cell membrane, and this sodium potassium ATPase, as you all know, it consumes around 20% of ATPs that are present in that are produced by our cells, 20% of the ATP. So that means that is a higher consumption of ATP by sodium potassium ATPase. So that means, so lot of energy need to be consumed. So that means metabolic rate has to go on at a higher rate to provide those ATPs, because oxidation of food material has to go on. So that means thyroid hormones, by increasing sodium potassium ATPase activity, it increases the basal metabolic rate. That is why in hyperthyroidism, whenever there is excess thyroid hormones, it is going to simply increase the basal metabolic rate and doesn't allow person to put on weight. So usually hyperthyroid patients, they are underweight. So now how to measure brown adipose tissue? Brown adipose tissue is measured by PET scanning or PET scanning, which is the gold standard imaging technique for brown adipose tissue. Now let me explain you briefly about reactive oxygen species. Now the reactive oxygen species are also referred as free radicals, and these are highly unstable products of molecular oxygen. Example, we have superoxide, hydrogen peroxide, hydroxyl radical. All these molecules, they have unpaired electrons in their outer orbit, or they are going to make a product that has got unpaired electrons in the outer orbit. So what will happen? So these free radicals, they are going to go and attack any molecule that are richly that are having electrons. If they can go and attack lipids, they can go and attack proteins. So if they attack lipids, so they are going to cause lipid peroxidation, means damage to the cell membrane. If they attack proteins, so they cause protein degeneration. If they attack DNA, so they can cause DNA damage. So overall, excess reactive oxygen species molecules can damage our biomolecules. So what are the conditions where excess reactive oxygen species can be generated? One of the condition where reactive oxygen species can be generated is the electron transport chain, especially electron transport chain that will run in a tissue which is partially oxygenated previously, and when sufficient oxygen containing blood is flowing through, so the gush of oxygen containing blood will lead to increased electron transport chain, and whenever there is increased electron transport chain, so there is a production of reactive oxygen species molecule, especially in ubiquinone, that whenever one proton is transported to ubiquinone, especially when it is a semi-ubiquinone form, so rather than ubiquinol form, that is 𝐒𝐞𝐻2, instead of 𝐒𝐞H is there, so that is one of the way to produce a reactive oxygen molecule. So electron transport chain itself is a source of reactive formation. So how to neutralize these reactive oxygen species? So there are something called as antioxidants. These antioxidants, they are rich in electrons, and so they are going to provide that additional extra electron that is necessary for reactive oxygen species molecule. So what is the example for antioxidants here? So antioxidants in our cells can be superoxide, it can be catalase, as well as it can be glutathione peroxidase, which is neutralizing hydrogen peroxide. It can be glutathione, which is used in the neutralization of hydrogen peroxide by glutathione peroxidase. It can be vitamin E, which is present over cell membrane, or vitamin C that is present in the cytoplasm. All these molecules, they are having an ability to donate that extra electron that is needed by reactive oxygen species and neutralize the molecules. Now let's move on to see what are the diseases that are affecting oxidative phosphorylation because of mutation in the mitochondrial DNA. Now let's move on to see what are the diseases that are involving oxidative phosphorylation because of mutation in the mitochondrial DNA. So the name of those disorders are 𝐒𝐮𝐬𝐜𝐫𝐢𝐚 syndrome, Pearson syndrome, MERRF, that is myoclonic epilepsy, ragged red muscle fibers, MELAS, that is mitochondrial encephalopathy, lactic acidosis, stroke-like episodes, Leigh syndrome, that is subacute necrotizing encephalopathy, and LHON, that is Leber hereditary optic neuropathy. In all of these disorders, there is an involvement of mitochondrial DNA mutation leading to defect in the oxidative phosphorylation, giving rise to variety of signs and symptoms. So that's all about inhibitors and uncouplers of electron transport chain, reactive oxygen species molecules, brown adipose tissue, and diseases that are related with oxidative phosphorylation because of mitochondrial DNA mutation. Thank you.